Dr. Kary Mullis won the Nobel Prize in Chemistry in 1993 for the development of a process that allows for amplification of DNA through an in vitro system. Since it is at the heart of DNA cloning and modern forensics, that award would seem to be well deserved. Polymerase chain reaction, or PCR, is a method for amplifying a small amount of DNA based on in vitro DNA synthesis. This is possible even for samples that contain a mixture of DNA molecules. Since the DNA is double-stranded, synthesis involves making multiple copies of both strands. The power of the process is the ability to repeat a series of 3 steps 20 to 30 times using a thermocycler to automate the process. First, the temperature of the reaction is raised in order to melt the DNA into a single-strand form. Next, the temperature is cooled in order to anneal the target DNA to a small DNA primer. Then the temperature is raised to the optimum temperature for catalytic activity. The DNA polymerase present in the mixture binds to the annealed primer and extends it to the full length of the template DNA. This process is outlined in the figure below. Please note that since there are 2 strands of complementary template, there must be 2 separate primers, one to anneal to the end of each template. Also note that the temperature is repeatedly raised and lowered with each cycle. These same 3 steps are repeated multiple times, and the amount of DNA is doubled with each step. By examining the figure, you can see that the primer in one step becomes a template in the next. Since the DNA is heated and cooled with each cycle, most DNA polymerase enzymes would be denatured by this process and unable to carry out synthesis. The key to the success of this method is the use of a heat-stable DNA polymerase. The first to be used was isolated from the thermophile Thermus aquaticus and is therefore called Taq polymerase. Functioning in an organism that grows at high temperature allows these DNA polymerases to tolerate the repeated cycles of heating and cooling without losing activity. There are many variations to these basic steps, but the principles employed are the same as outlined here.